This is a presentation for the poster Missense 3D. How effectively can one model the structural impact of a missense variant in both experimental and predicted globular and transmembrane structures? The presenter is Mike Sternberg from Imperial College London. It's well known that by examining a protein structure, one can interpret the impact of a missense variant, as illustrated in the slide, where the wild-type His-glue salt bridge is abolished when the His becomes a tire mutant, and this leads to a disease. Of course, one requires structure for such analysis, and our group has developed and exterminated the Phyre protein structure prediction server, which has over 11,000 citations and more than 2 million submissions. When applied to the residues in the human proteome, we see that experimental PDB structures would cover 13%. However, predicted structures add an additional 33%, and so totally one would be able to get just under half of the human proteome from globular proteins, predicted and experimental. Accordingly, many of our Phyre users contact us and ask us if they can use the predicted structure accurately to interpret the effect of a genetic variant. This question motivated us to develop missense 3D, where we would examine in detail the structural impact of a missense variant. So the methodology starts by locating the variant, identifying the local region, removing the side chains, mutating the residue, and repacking the side chains. Then we would examine in detail the impact of these changes between the wild type and the predicted structure, for example, asking if a disulfide bond is broken or if a proline residue is introduced. Importantly, we use soft features for evaluating these effects. So, for example, disulfide bridges, hydrogen bonds, and salt bridges, we increased the required distance by 1 angstrom to allow for any errors in the predicted structure. The procedure has been extensively benchmarked, and we have used data from Humsavar, Klinvar, and Exact of disease-associated and neutral variants. And initially we started with structures from the mole probity data set and evaluated how missense 3D would work on that. We then predicted Phyre structures for these proteins and re-evaluated it. Back on the PDB structures, we see we have a true positive rate of 40% and a false positive rate of 11%. Importantly, features like disulfide bond breakage, the introduction of a buried proline, introduction of a clash, and changing charged residues into the middle of a protein have a high specificity, enabling us to have a far higher true positive rate than false positive rate. We then proceeded to evaluate the same variants on predicted structures with a back-to-back evaluation of the Phyre structure and the crystal structure. And you can see on the next slide how that the Phyre structures with the true positive and the false positive rates are comparable to that of the PDB structures. Missense 3D is available as a web server to the community, and you can input your variants either from a UniProt location or via a PDB location, and you can also upload your own protein structures. Additional resources closely integrated to missense 3D is our Phyre risk database, where you can input genetic variants either from a genomic or a proteomic coordinate set, and these will be mapped onto predicted or experimental protein structures. and then one can go and use missense 3D for detailed evaluation. Work in progress is our development of missense 3D DB, which will be a database of structure-based annotations for 5 million missense variants from Humsava, Clinvar and Nomad mapped onto PDB and Phyre predicted structures. A further development is to extend missense 3D to consider variants within the transmembrane region of proteins. The location of the transmembrane protein within the lipid is modelled. The algorithm, for example, penalises charged and polar residues exposed to lipid. On a test set, the TPR was 44% and the FPR 10%. To summarise, missense 3D can provide structural explanations for missense variants in both experimental and predicted structures. It's available as a web server at the following address. Missense 3D provides structural annotations for 5 million variants, and we are also extending missense 3D to model variants within transmembrane regions. The next slide summarises the 3 key references, and I thank you for your attention and acknowledge the contribution of the team.